Hi everybody, welcome to the Characteristics of Life in Chemistry Crash Course video. That was weird saying, but this is going to help you with your study guide. Remember that the things that are highlighted are extra credit. You can either print it out or you can type it in straight into the document. Once again, as I've been saying, do not use this as your only form of studying. This is just to help you understand the main points and to support you with the study guide. You should be using your other assignments and the other video lectures which go more into detail. This is just a supplement. So let's begin with the characteristics of life. There are 7 characteristics of life that we talked about in class, 9 if you break these down into 2 different groups. But that first characteristic of life that we talked about is cells in an organization. What we really mean is living things have cells and are well organized. Second characteristic of life is energy processing and metabolism, meaning that living things use energy. And because we use energy, we need to have a way to break down that energy. So we talked about that in cellular respiration. And recall that the definition of metabolism is not just about how much you eat and how much weight you gain. It is talking about the sum of all the chemical reactions in your body. Our third characteristic of life is reproduction with heredity. Make sure that if you answer with this, you include the with heredity, because a fire can reproduce, but that doesn't mean it's alive, okay? That heredity is when you pass down that genetic information from the parent to the offspring, and that is how we reproduce. But recall that there are different types of reproduction. There's asexual, where it's just yourself, or sexual, where there is a partner. Homeostasis is how the body stays alive. Many causes of deaths are failure of homeostasis, and homeostasis is just how your body self-regulates itself. Okay? Your body uses a combination of different mechanisms to maintain homeostasis. If you didn't have a sense of homeostasis, you wouldn't be a living thing, and you'd probably be dead. Growth and development is another characteristic of life. This is just saying that living things will grow over time, and they will develop. Now, living things have different growth and development patterns, so we're not saying that there's a specific pattern that needs to be followed. We're just saying that living things will do that, right? Because seeds develop differently than we do as babies. A response to stimuli just means that things that are living are aware of what's around them and can react to things that are near them. Now, these don't have to happen instantaneously, right? Like with plants, they will react to the environment around them. When it snows, they will start to go dormant. If it rains, they will increase the rate of photosynthesis. And our last characteristic of life is regulatory mechanisms, which just means that the body or that living thing has a way of maintaining homeostasis. The specific ones that we talk about are gas exchange and water regulation. So for us, gas exchange means breathing, and water regulation means urinating and drinking water, but don't drink your urine. Okay, moving on to macromolecules, the chemistry aspect of this. The macromolecules that we talked about were carbohydrates. The building blocks of carbohydrates are monosaccharides. These technically count as a monomer, and a monomer is just one part, right? And a polymer, which is the carbohydrate, is many parts of a monosaccharide. So this is the monomer, this is the polymer. Some examples of carbohydrates are sugars and starches, right? These are the things that you eat. If you're on the keto diet, you will avoid carbohydrates, so you'll avoid bread and sugary things. The functions of carbohydrates are to store energy and provide structural support. That makes sense, right? Because glucose, as you know, is a product produced in photosynthesis that is very good at storing energy. The plant produces glucose because it can't just store ATP, because ATP is very energetic and unstable, so it transforms it into glucose, which is a carbohydrate. So you already know that. Moving on to lipids. The building blocks of lipids are fatty acids and glycerol. There is no monomer because these are 2 different units that build up lipids, so there is no monomer of lipids. Some of examples of lipids are fats and oils. These fall into like butter, right? The oil that you use when you're cooking. And the function of lipids are to store energy and provide structure. Lipids store energy better than carbohydrates do. That's the key, right? So lipids and carbohydrates have 2 of the same functions, but lipids store energy more efficiently than carbohydrates. Moving on to nucleic acids. You might recognize that word nucleic acid from DNA, deoxyribonucleic acid. The building blocks of nucleic acid are nucleotides. This is a monomer because it is a repeating unit that makes up the polymer of nucleic acid. Examples of nucleic acids are DNA and RNA. And the only function that nucleic acids have is to store genetic information. Okay, that is one unique function. No other macromolecule does this. It is only nucleic acid that stores genetic information. Finally, my favorite, proteins. The building block of proteins are amino acid chains. Okay, you don't have to know the amino acids, but know that they're made up of amino acids, and there are different types of amino acids. The structure of a protein is dependent on the amino acid chains. Because there are different types of amino acids, you can string them together in different ways. This is going to change the shape, and the shape of the protein determines the function. Proteins have 4 different functions. That's why they are so very important to our daily lives. They will speed up reactions. We will talk more about enzymes in a little bit. They provide structural support. This is familiar, right? We saw this in lipids and carbohydrates. They send signals. So a lot of the signals that our body sends, like, hey, you're hungry, or hey, like, you need to produce more of this, or something needs to happen, those are facilitated through proteins. And they will transport materials. Hemoglobin, your blood, will transport oxygen and carbon dioxide throughout your body. That is done through a protein. Okay, we just talked about proteins. Proteins are enzymes, but not all enzymes are proteins, because enzymes are specialized proteins that will speed up reactions. Okay. This is how enzymes work. A substrate, something that needs to be reacted on, okay? This is a reactant. It will go into the active site of an enzyme. Remember that enzymes are pretty specific. So if this substrate did not fit the shape of this active site, it'd be like, nah, you're not welcome here. When that substrate binds to the active site of the enzyme will make it a little bit more comfortable, right? We talked about this in class, how when you're more comfortable, you will get more work done, hopefully. So when this enzyme makes this substrate comfortable, a chemical reaction can occur. However, there are a few things that can happen if this active site is a little bit denatured. And when we say denatured, we're saying that the amino acids, the chain of amino acids in this has been a little bit altered. So this active site no longer has this same shape. It'll look like a flat line, or just like completely a curve. That's what we mean when we say denatured. The enzyme is not broken, it's just lost its biological function. If that happens, then this active site can't make this substrate comfortable and no reaction will occur. However, this active site is not denatured. It is still good. It's still a regular enzyme. So it can continue to make products. When that happens, when that enzyme makes that active site really comfy for the substrate, the enzyme and the products will form a complex. The enzyme will do some work and products will be released. Then this active site will once again be able to take in another substrate. So we talked about how this active site can get denatured, and a lot of things can do that. So things like high acidity, right? So high or low pH, really high temperatures, really low temperatures. There's a lot of things that can denature it. There's pressure differences that can happen. We didn't really talk about that, but this active site can get denatured. Refer back to your notes if you want more information. However, just as it can get denatured, we can increase the rate of this reaction. We can increase the rate of product formation. If we have more substrate, we'll have more products. That will increase the rate of product formation. If we have more enzymes, there will be more enzymes available to take on substrate. So that in turn will also speed up enzyme activity and increase the rate of that reaction. All right. Good luck studying, everybody, and I hope you all have a wonderful day.